Hi, this is Joe Ross from Fresno State, and I'd like to take this opportunity, although I'm not there in person with you, to explain my lab's research into how genetic differences between parents can cause hybrid dysfunction, that is, lethality or sterility, in their offspring. And this is the biological species concept by Ernst Mayr, that when you cross two parents, if they're from different species, their offspring are either infertile or they're inviable. Both of those conditions are caused by developmental genetic defects. Some combination of alleles from the two parents brought together in one organism causes that developmental dysfunction that causes death or sterility. And we're trying to figure out where those mutations exist and what their identities are. And this Dobzhansky-Muller incompatibility model can help us understand how those sorts of mutations might arise. So we have two individuals in a population, and they can reproduce and produce more offspring of similar fitness. If this population gets divided somehow into two distinct populations, those populations can accumulate different mutations that are neutral within those populations. So we continue to have production and fit individuals. But if at some point in the future those two mutations are brought together in one organism, those two mutations at that point can negatively interact to cause hybrid dysfunction. If these hybrids here are sterile or inviable, that would indicate that speciation is complete. But it's possible that these mutations create less fit hybrids, and that might indicate the onset of speciation. And today I'm going to tell you some empirical evidence that we have that two populations of C. briggsae, HK104, blue, and AF16, red, do exhibit this sort of property, that they produce hybrids that are less fit but not totally unfit. And so we use C. briggsae to study the onset of speciation. And the reason that we study C. briggsae is that they exhibit some interesting patterns of genetic diversity that C. elegans, the sister species, doesn't exhibit. And also, as I just mentioned, we have this empirical evidence of hybrid less function. So this is a phylogenetic tree of C. briggsae wild isolates, and I'll point out that there's a group of tropical clades here and some temperate strains and clades here. I'll focus again on AF16, an Indian strain that comes from a region of India with a temperature of about 25 degrees on average, and a Japanese strain, HK104, average temperature about 20 degrees. And this partitioning of the tropical strains and the temperate strains suggests that there's some fixed genetic differences between the temperate and tropical clades that might have something to do with temperature adaptation. And most work in C. briggsae and C. elegans occurs at 20 degrees, and what I'm going to tell you about today is an experiment we did first at 20 degrees, and then at 25 degrees to address the question of whether or not there might be genetic differences between these two clades that predispose them to optimal life at those two different temperatures. And we do that by creating hybrids and then looking at whether or not those AF16 HK104 hybrids exhibit a phenotypic defect like inviability or infertility, but also whether or not there are particular combinations of alleles in their genomes that are incompatible with each other. So we create advanced intercross recombinant inbred lines, and in this case we have a hermaphrodite and a male, one from AF16, red chromosomes, and HK104, blue chromosomes. We've got an autosome pair here, an X chromosome pair, and the mitochondrial genotype from the hermaphrodite and the male. They're totally homozygous throughout the nuclear genome. So in the F1 we have a totally heterozygous nuclear genome. We SIB-mate for 7 generations, and then we self for 10 to create advanced intercross recombinant inbred lines that are totally homozygous at each locus with a random, essentially, haplotype block patterns throughout the nuclear genome. And we've created about 167 of these, genotyped at over 1,000 SNPs throughout the genome. And this was done first in our 2011 paper at 20 degrees. And the way we're going to represent the genotypes is by looking at marker transmission ratio distortion. And that is there a preferential inheritance when these hybrids are grown at 20 degrees of perhaps HK104 or temperate alleles. So this is just like that recombinant inbred line cross. We have totally heterozygous F1 individuals crossed together. And what we're going to suggest in this cartoon is that there might be a genetic interaction, negative interaction, between homozygosity for AF16 in the middle of this chromosome and being heterozygous elsewhere in the genome. If that is an unfit combination of genotypes, homozygous red versus, say, heterozygous over here, or on a separate chromosome, then when we get the ultimate F17 generation recombinant inbred lines, there will be underrepresentation of red alleles in the middles of these chromosomes, and it can be plotted as marker transmission ratio distortion, where we have a plot of fraction of all 8 of these lines, strains, containing the blue allele, or HK104. So 100% blue is up here, and we see that in the middle of the chromosome, where we lost, by selection, red alleles in an earlier generation, there's 100% blue genotypes here, where we expect, because we started in the F1 generation with 50% blue, that's our expected value, and that's what we observe on the edges of the chromosomes here. So deviations from the expected value indicate that there's a region of that chromosome that's potentially participating in an incompatibility with a locus elsewhere in the genome. And these are the actual data from those 167 recombinant inbred lines at 20 degrees. And there's some interesting patterns here, but the most salient is that the 50% line, this thin black horizontal line, was the expected value for these crosses. And we observed that on most of the chromosomes, especially chromosomes 3, 4, and 5, there's a bias, and the bias is usually toward inheritance of blue or HK104 alleles over red alleles, AF16. And that's why we came up with this hypothesis that maybe HK104 alleles in general make hybrids more healthy when they're grown at a temperate or HK104-like temperature. And part of this might have to do with coadaptation between the mitochondrial genotypes and the nuclear genotypes of these worms. And there could be some compensatory mutations between the mitotypes and the genotypes of the nuclear alleles that would cause those sorts of patterns. And one way to think about this is that if you have blue or HK104 nuclear and mitochondrial alleles, gene A and gene B, their protein products might fit well and function well together to produce ATP and to produce phenotypically fit offspring. And if you have a mismatch in those protein shapes or structures, their interactions might lessen the efficiency of electron transport and cause some sort of hybrid dysfunction. And so our hypothesis, again, was that instead of seeing at 20 degrees a bias towards HK104 alleles, perhaps when we do this at 25 degrees C, a more AF16 or red-like temperature, maybe we would see a bias towards 0 HK104 inheritance, or bias towards then AF16 inheritance, a dip below 50% in these plots. And my graduate student, Rania, who did all of this work, created a number of recombinant inbred lines using the same cross design, but instead at 25 degrees instead of at 20 degrees. And what she's done is she's looked at the 6 different chromosomes in C. briggsae, and she's just spot genotyped them so far at a few single nucleotide polymorphisms we can easily genotype throughout the genome. And this is a similar sort of allele fraction plot where 100% blue alleles is up here and inheritance of only red or AF16 alleles is here. The dashed lines are expected frequencies for the autosomes and for the X chromosome. And anywhere we see a bracket indicates statistical difference between the observed and the expected allele frequencies. The blue symbols are individuals that have HK104, blue, temperate-like mitochondria, and the red circles are the AF16 mitochondria. There are only 2 chromosomes which we saw biases. One is chromosome 3, and chromosome 3 we already suspected we would observe this because we know that inheritance of chromosome 3 AF16 alleles in C. briggsae hybrids causes developmental delay, which is a separate genetic interaction, again, we already knew about. What we were not expecting, though, was this interesting bias on the X chromosome, where if you have HK104 mitochondria across much of the X chromosome, HK104 mitochondria is coupled with not inheritance of blue X chromosomes, but there's a bias towards inheritance of AF16 or tropical-like at 25 degrees growth, tropical-like X chromosomes. And so that is the basis for the title of the talk. If you can't stand the heat, get your X to the tropics. It seems like the AF16 X chromosome has potentially some genetic variants that are beneficial when growing at a tropical or 25 degree temperature. And that could occur because of transmission bias, that is, for HK104 mitochondria together with AF16 X chromosomes might also result from deleterious interactions between red or AF16 mitochondria and blue or HK104 X chromosome alleles. And that harkens back to that mitonuclear coadaptation model, where if there are genetic variants that are unfit with this combination of alleles, then that would create a bias or an apparent bias towards HK104 mitochondria and AF16 X chromosome inheritance. So in the future, what we're going to do is look beyond just AF16 HK104 hybrids to make more hybrids of tropical and temperate strains to see if this pattern is more pervasive than just between these two specific strains. I'd like to thank again Rania, who did all of this work, some undergraduates and technicians in the lab who supported this work, and also funding sources from the California State University system. Finally, if you know anybody who wants to be a microbiologist at Fresno State, please give them this information and refer them to us. Thank you for your time.